Okay. Okay. I think we'll get started. It is my pleasure to introduce Holly Holmes today for her proposal defense. Holly, if you are joining us on Zoom, thank you for joining us. We're excited to have such a great Zoom audience, and please, to the extent that you are able, keep an eye on keeping your mute buttons on and your video off for the time being, so that we don't run into bandwidth issues here at the university. But if we need to address something in the talk, we will do that so that, yeah, things don't get disrupted. My name is Hannah. I am a research scientist here at the University of Montana, and I am proudly Holly's co-advisor along with Josh Millspaugh. I want to say thanks ahead of time to Holly's committee, who is all present either on Zoom or in the room here, and acknowledge up front that this project is coming to us from Glacier National Park, from one of Holly's committee members, Lisa Bate, who has worked on Harlequin Ducks for a long time, and it's the product of a big collaboration. So there are a lot of folks involved in this project, and Holly will go into that during her talk. Holly is most directly joining us from work at Glacier. She spent 5 seasons working there, and she's worked on all manner of critters. But she's also worked in a number of other parts of the West. Between her undergrad, which she did at Colorado State University, finishing up in 2019. And she actually hails from Colorado, so that's a life in the Rocky Mountains is one that is familiar to her. And though she has worked on all manner of critters and all manner of places, I think I've come to really appreciate about Holly that mountain associated critters are a bit of a part of who she is. And she relates very well to them. If you wander around in the mountains of Western Montana over the last 5 years, it would not be unlikely to run into Holly somewhere out on a trail. So if you're looking for her outside of work, that's probably where you can find her. One of the other things that she's mentioned is that she has a real passion for collaborative conservation and place-based conservation. And I think that this project is really well suited for Holly because it is a big collaborative project, and it really does pay a lot of attention to place. So we're really excited to have Holly's enthusiasm and her expertise working on this very cool, very cool species that we really have a lot of information needs about. So we're excited to hear your proposal and excited that you've joined us to work on this project. Take it away. Thank you. But we are going to turn the lights down, Holly.
That sounds good. There's no intermediate lights, so we'll just be in darkness for this presentation, but you can see my presentation. Yeah, thank you. Thank you for that introduction, Hannah, and thank you everyone here in person and on Zoom for attending my proposal defense. It means a lot to me that you're all here. So this is my proposal defense on investigating non-invasive survey methods for studying Harlequin ducks on their breeding grounds in the Northern Rockies. So before I get started, I want to introduce you to who is the Harlequin duck, especially for those of you who may be less familiar with this species. The Harlequin duck is a small, long-lived, and slow reproducing sea duck. So on average, they live about 15 years in the wild. The oldest recorded Harlequin duck was 20 years and 9 months old, and they don't reach sexual maturity until around the age of 2. However, their reproductive success stays low until roughly around the age of 5. There's no other species that's really closely resembles the Harlequin duck, and they are characterized by the male's coloration, which is this beautiful burgundy, slate blue, black, and white coloration. And the name Harlequin actually originates from a character in 17th century French passion plays, the Harlequin, which was also characterized by its burgundy and black costume. They're sometimes referred to as sea mice for their characteristically unduck-like squeaks that they make. And there are 2 distinct populations of Harlequin ducks in the world. There's one in the Atlantic Ocean and one in the Pacific population. For my specific study, I will be specifically focusing on the Pacific population, and even more specifically, the population that breeds here in Western Montana and Northern Idaho. And so this population of Harlequin ducks, like I said, they are a sea duck. So they winter along rocky coastal shorelines up the coast of British Columbia, up into Alaska, into the Arctic and subarctic. And then they exhibit a really unique east to west migration where they migrate inland to breed on fast moving whitewater streams in the mountains. And so this leads me to my next point, that they're kind of like your most hardcore raft friend because they are immensely strong swimmers and they're whitewater specialists. They can hold their breaths up to 45 seconds and they can dive up to 70 feet to forage on microinvertebrates that they feed on. Also, a fun fact about Harlequin ducks is they actually have more broken bones than any other species, and adults often live with multiple healed fractures. And then further adding to my point about them being your most hardcore raft friend is they're more strictly confined to running water than any other waterfowl species in the Northern Hemisphere. So this means that on their breeding grounds, we find them on streams, rivers, and creeks, and not boring still water like lakes and ponds. However, despite being incredibly tough, biologists have become increasingly concerned about the status of Harlequin duck populations. And so this is a graph showing counts from a long-term study that's been going on in Glacier National Park, where every year they go out and they do 10 ground-based foot surveys to count Harlequin duck breeding pairs on the streams, breeding pairs and broods. And so on the x-axis, we can see the year, and the y-axis is the high count from each of those 10 surveys throughout the year, with the trend line showing the decline. And apart from just Glacier National Park, there's also other long-term studies going on, such as in Banff National Park, there's one outside of Jasper in Alberta, and there's one on the Lochsa River in Idaho, which have all documented similar declines. And apart from just their breeding grounds, we've also been documenting declines on wintering ground areas such as Hornby Island, which is off the coast of British Columbia. And so in 2001, the Atlantic population of Harlequin ducks was listed as a species of special concern in Canada by the Committee on the Statute of Endangered Wildlife in Canada. For those of you that may not be familiar with that is, that's similar to our Endangered Species Act here in the United States. This is a screenshot from listserv showing Harlequin ducks status in each state or province. And as you can see in the lower 48, they're considered imperiled or critically imperiled, depending on the state. In Canada, they're considered vulnerable, and the only place they seem to be secure is in Alaska, which seems to be the last stronghold of Harlequin ducks. And so why are they declining? I think to understand this, or begin to ask this question, I think we need to understand just a little bit more about their life history. So Harlequin ducks, they are a monogamous species, and they'll form their pair bonds on their wintering grounds around the age of 2. And then after they form their pair bonds, they will migrate to the female's natal stream. And so the males will follow the females to their natal stream, like I said. Now, this is a graph, or this is a map that comes from a study that was done between 2016 and 2019, where biologists placed geolocators on Harlequin ducks to learn about their movement and their migration ecology. I think it's just a really cool representation of their east to west migration that is unique to Harlequin ducks. However, like I said, the Harlequin duck, so the females are extremely site loyal, and they'll return back to the stream that they were born on every year to have their own. And so when they arrive on their breeding streams, the females will the males will stick around for about 1 month and mate. And after they mate, the males will migrate back while the females lay. This is just a really cool representation of, it's data from eBird, where people upload observations of Harlequin, like visual of looking at, if you watch here in Montana, we see this pulse of Harlequin ducks when the males arrive, and then it dies down. And then we see another pulse when the chicks are born, and it dies down mostly because So like I said, the males will leave after about a month, leaving the females to incubate their eggs and to raise the young on their own. And so this behavior of males leaving early creates this irregular levels of reproductive success for breeding Harlequin ducks, and that's because if one nest fails, there's no chance at a second clutch because they left. Okay. And so because there's no chance at a second clutch, climate change impacts on stream flow to Harlequin ducks can be especially harmful. So I think this is a really cool visual of that. This comes from a paper Warren et al. 2019, who was actually another graduate student here at UM who studied Harlequin ducks. But I like it because we can see on the x-axis, it's the month of the year, and the y-axis is cubic feet per second. So the runoff of streams and rivers. And then we see the different breeding time periods. And if we can imagine this graph shifting to that's the right, later in the year, we see the peak stream flow happening in the incubation, washing out nests and eggs. However, if we see the opposite of that happening, where this figure in the peak stream flow is happening earlier in the year, then during the hatching season, when they're reliant on there being water in the streams, and we see streams drying up. So these climate change impacts, either direction, whether it's extreme flooding or extreme drought, both have pretty detrimental effects to breeding Harlequin ducks. And apart from just the climate change effects, there's also anthropogenic threats that are affecting Harlequin ducks. So these include things like the tremendous increase in river-based recreation. Summers are getting hotter, people want to spend more and more time on the water, as well as increase in encroachment from human development, which leads to habitat loss. And so because of all of these, because of their strong site fidelity, their irregular levels of reproductive success, and their slow maturity, it's thought that breeding streams have a really low likelihood of being recolonized once all of the breeding pairs on a stream are lost. And so traditionally, we've relied on these direct in-person ground-based foot surveys to survey for Harlequin ducks. Now, these ground-based foot surveys, they are pretty limited in the information they provide because of conditions that are outside the observer's control. Conditions include things like the spring flooding that happens every year, where birds might seek backwater refuges. Also, when there's this spring flooding, it can be really unsafe for people to be in or near waterways. And so doing these surveys becomes even more challenging as we're trying to observe them from a safe distance. Also, just from personal experience, these surveys are pretty challenging to conduct because you're either slipping and sliding for many miles up rocky stream beds, or you're bushwhacking through riparian brush for those same many miles. And so, independent of the detection process, spring flooding can also affect stream occupancy if nests are washed out and females leave the area earlier than if their nests were successful. So using these current ground-based foot surveys, it's really hard to know if the lack of observations is bird abundance related or observation process related. And then furthermore, no one's ever figured out what the detection probability of doing a single one of these ground-based foot surveys are, and oftentimes a stream might only get surveyed once every 5 years. And so I've been working with biologists across the Northern Rockies here in Montana and Idaho, Alberta, and Wyoming. And it's become clear that there's an interest and a need to attain, one, a deeper understanding of this survey method that we already have, the ground-based foot surveys, but also to explore other detection strategies and tools that might help us improve our understanding of Harlequin ducks and their occurrence and abundance patterns. And so this leads me to my thesis. So for my project, I'm proposing 2 chapters. The first will be focused on investigating the efficacy of new non-invasive methods and detection strategies for detecting occupancy state on a stream. And my second chapter, I aim to build a predictive occupancy model that can help identify areas of potential breeding of Harlequin duck in Western Montana and Northern Idaho. So in a nutshell, we're looking at how can we find where these birds are, and then where are they, to use these methods. Okay, so this leads me to my first chapter, investigating non-invasive survey detection strategies. Okay, so non-invasive survey methods are becoming more and more popular in wildlife and conservation research. And so essentially what they are is just ways to survey for animals without causing harm or stress to the animals. So we're not physically handling these birds, but we're trying to detect them in other methods, with other methods. And so the 4 methods that we're looking at are these ground-based foot surveys, environmental DNA or eDNA, motion detection game cameras, and time-lapse game cameras. So I've already touched on ground-based foot surveys a little bit, but essentially they are what they sound like. It's teams of observers walking up a stream looking for Harlequin ducks. And the main thing that's important about these ground-based foot surveys is that people are working in teams of at least 2 to ensure that one person always has their eyes on the creek to make sure that no duck is missed or double counted as they're moving up and down the stream. So environmental DNA is our next method. This is also the method that I probably get the most questions about, and it's the method that I'm working and learning the most through, maybe not the most, but I'm working and learning a lot about it as we go. So how does it work? So DNA comes from anything that's sloughed off an animal that has cells. So this could be saliva, feces, feathers, blood, dander, etc. And then an environmental sample is essentially what it sounds like. So that could be things like a water sample, snow samples, a swab off of a plant, or even air samples, which have been used for DNA. So for our case, we're using water samples to look for Harlequin duck DNA. And so because Harlequins are strictly confined to running water and water is running, we thought that this could be a good opportunity for the river to almost act like a conveyor belt of DNA if we could detect Harlequin ducks farther upstream. And so one important note about eDNA, because I do get this question sometimes, is it can only tell us presence or not detected. Unfortunately, it can't tell us abundance. It'd be really awesome if it was like a 23andMe and we could sort things out, but unfortunately, right now, it just tells us presence, they are here, or they were not detected. And then my 3rd and 4th methods are game cameras. And so game cameras are great because they allow for detection of species that would not be seen using other methods due to animal behavior or human error. However, game cameras are not typically used on running water or on waterfowl. And that's because they rely on, well, motion detection ones rely on motion detection. So if you're placing a camera on running water, it might just trigger the camera a lot. Also, waterfowl are extremely insulated, so they have a really small heat signature, and they're a much smaller just body size in general to mammals, which game cameras are normally used on. However, Harlequin ducks, they exhibit this life history trait where during the females, like the brood rearing season and their nesting season, they tend to stay within about a 1 mile reach of stream where they'll forage downstream and they'll fly or swim back upstream for roughly about 1 mile. And so because of this behavior, we thought that, well, maybe if we place game cameras every river mile, we might be able to detect Harlequin ducks. But again, because they're not typically used on running water or on waterfowl, we wanted to test both motion detection and time lapse to see if one of these methods would work for detecting Harlequin ducks. Okay. So the objective of this chapter is to assess the efficacy of 4 different methods for detecting Harlequin ducks to determine what detection strategy or combination of detection strategies has the highest probability for detecting Harlequin ducks on a stream given variation in the habitat covariates. And so we surveyed 10 streams across Montana and Idaho in both 2022 and 2023. So each of these sites was surveyed 4 times, more or less. There was a couple that got missed because of flooding or fire. But these streams are representative of a variety of ecosystem and stream conditions used by Harlequin ducks for breeding. Each of our study sites was deliberately chosen because they've historically had breeding Harlequin ducks and they've had confirmed breeding in the last 5 to 10 years. We also intentionally selected streams with high probable occupancies so that we can have greater power with a smaller sample size to look at what drives Harlequin duck detections. So streams that were chosen that exhibited variation in stream flow patterns, elevation, and vegetation cover. And the timing of the site visits was dependent on the stream's elevation. This is because elevation has been shown to affect Harlequin duck breeding phenology. So ducks at higher elevation streams tend to breed and migrate later than ducks at lower elevations. And like I said, we visited all of these sites 2 times in both seasons. So the first site visit happened when we would expect to see single females who were either just still nesting or broods that just hatched. So just super fluffy down fluff balls. So this was early July through early August. And the second visit happened about 3 weeks later when we would expect to see females with their broods. And so this was late July through late August, depending on the elevation of the stream. And the reason why we decided to visit each of these streams at 2 different time periods is because we think that the detection of Harlequin ducks during these time periods is going to differ. Okay, and so, yeah, so each site visit, every time we visited these streams, each site visit consisted of 3 days. And on all 3 days, we did a ground-based foot survey. And on the first day, we did a ground-based foot survey and put up game cameras. On the second day, we did a ground-based foot survey and took eDNA. And on the third day, we just did a ground-based foot survey. And the reason for doing 3 ground-based foot surveys is we wanted to have repeat observations of these foot surveys within an area where we were assuming closure of the population. And so we were not expecting the population to change a lot in these 3 days. And so in 2022 and 2023, we were able to conduct 118 ground-based foot surveys, 723 eDNA samples were collected, and we placed 198 game cameras, half of those being time-lapse and half of those being motion detection. And so what are we going to do with all of this data? So I can confirm that Harlequin ducks are present on all of my streams by looking across all 4 of my methods. And as long as Harlequin ducks were picked up in one of my methods, I can say that they're present. And so because of that, I can use logistic regression models to estimate my detection probabilities. But to use the logistic regression, I'm going to need to set up my data so that I have discrete detection, non-detection data. And so to do that, my unit of replication for the ground-based surveys will just be 1 survey, as well as for the eDNA, it'll just be 1 eDNA sample and 1 camera with both game time-lapse and motion detection considered separately. And then variation in the effort across each of these samples will be addressed using covariates in my model. And also each model will include a random effect, which will take into account some of the heterogeneity between the streams that we weren't able to measure. And each model will also be fitted with a link function. And so I plan on building 4 main models that will help describe the process of how habitat covariates that were collected while we were surveying. These are these on the slide, like the segment length, stream flow, segment character. Those are all my habitat covariates, and they were collected. And I'm going to look at how each of these affect with each detection method. However, because I am visiting each stream during both the nesting season and the brood rearing season, and I think the detection may differ between the 2 time periods, I'm actually going to build these same 4 models, but for both time periods. So at the end, I'll have 8 models instead of just 4. And then I'm going to use the model relationships to derive 2 main things. 1, the detection probability of each method at specific covariate values, and 2, the number of replicates of each method required to attain 80% confidence in detecting Harlequin duck, given that they are present on a stream. And then through understanding the relationship between these covariates and the detection methods, I can then begin to consider different scenarios of interest. For example, this would be like conducting a ground-based foot survey and taking eDNA samples, or taking many eDNA samples at a beginning of a stream versus spread out. Okay. So this brings me back to my proposed chapters. So like I said, the first chapter, we're really going to be focused on looking at these new methods of how can we detect Harlequin ducks. And then my second chapter is going to be looking at where are Harlequin ducks in the Northern Rockies. Okay, chapter 2, predicting occupancy in the Northern Rockies. And so working with non-game biologists, it's become clear that they're concerned about not fully understanding the scope of where Harlequin ducks are breeding within the region. In Western Montana alone, there are roughly 3,000 streams. And so it would be impossible for biologists to go out and survey all 3,000 of those streams. And that's just Western Montana alone. That's not including Idaho or Washington, or I mean, Wyoming or Washington, or the rest of their breeding range. And so managers have expressed information goals that include understanding where are populations of Harlequin ducks that we might not be looking that are potentially important. And so the objective of this chapter is to build a predictive occupancy model of breeding Harlequin ducks in Western Montana and Northern Idaho. To do this, I'll be gathering all of the data of evidence of breeding Harlequin ducks in Montana and Idaho over the last 15 years. I'll then be gathering remotely sensed habitat data for the region. And finally, I'll be building my predictive occupancy model by using an integrated spatial distribution model. And I'll be walking us through these steps for the rest of this presentation. So my study area, it ranges from the northeast corner of Glacier National Park on the Montana-Canada border, south along the Rocky Mountain front to the Wyoming-Montana border near Gardner and Yellowstone National Park, and then west through Idaho to the Idaho-Washington border. And now the points that you're seeing on this map, this is some of the historic data of breeding Harlequin ducks that we have in the state of Montana. There's also similar data for Idaho. I just haven't received it yet, which is why it's not on this map. And I'll be explaining where all of this data comes from in my next slide. But furthermore, within this region, I'll be focusing on Harlequin duck breeding habitat specifically. So this is streams, rivers, and creeks, like I mentioned before. I'll also be using other variables to further constrain my breeding habitat. And so some of these variables could be like stream order. So for example, we know that Harlequin ducks don't breed on higher order streams, such as the Clark Fork. It's not their habitat. We also know that they're not alpine species, so we can use variables like elevation to constrain above tree line is not Harlequin duck habitat. I'm also investigating whether I can use the Cornell Lab of Ornithology's American Dipper distribution model, because American dippers are an extensively studied species. And as far as we know, there's no habitat that's American dipper habitat that's not also Harlequin duck habitat. And so it could provide some good insight on different habitat to include or not include within this model, combined with the stream order and elevation. Okay, so we have Harlequin duck observation data going back to the 1860s in Montana. However, a lot of this data has been collected in different ways with different challenges and assumptions connected with them. So we have data from structured ground-based foot surveys that have been done by numerous different agencies. We have incidental data that's come to us from people who have just seen Harlequin ducks and reported it to these different agencies or to the Montana Natural Heritage Program. We have citizen science data that comes from platforms like eBird and iNaturalist. And there's also just a chunk of data that comes from a whitewater kayaker who just really loved Harlequin ducks. And so I've been thinking about how can we leverage all of these different types of data to help us address this information need of where are Harlequin ducks that we're not looking. And so the first step is I'm going to need to take all of this data that I'm getting from both Montana and Idaho and sort it into these different data types in ways that I can use all of the data that we have. And so these three different data types are as listed here. So there's repeated within season data. So that's data that comes from these structured ground-based foot surveys where we have multiple surveys done on the same site within a season. And this also includes citizen science data where there are multiple complete checklists within the same site within a season. Data type 2 is annual presence only data. And so this is data that comes from the structured surveys where there was only one survey done on a stream, or this is the incidental data, and much of the other citizen science data falls into this. And then the absence only data, this is augmented zeros from sites where we're confident do not have breeding Harlequin ducks. So going back to how I was talking about selecting our streams, there's certain habitat that we just know is not Harlequin duck habitat. And so for those areas, we can augment zeros into our model to give our models both the important zeros and ones that it needs to predict occupancy. Okay, and then I will be using an integrated spatial distribution model. And these models, they can allow us to predict occupancy by combining both the existing observational data that comes from the variety of sources and then also the incidental data. And so specifically, I will be adapting an integrated spatial distribution model that was discussed by Strobel et al. In 2022. And their model, it accounts for imperfect detection by estimating detection probability and detection non-detection data and observational data by using an occupancy model, while making use of the incidental data to inform the latent state. And so I've decided to predict occupancy because of the scale of interest that we have. The scale of interest that we're looking at are 1,600 meter reaches, so that river mile. And that's because it corresponds to the Harlequin ducks life history trait where they spend their given day within a 1 mile reach of stream. And because we're predicting it at that scale, most of the time we're probably only going to get one single female or one breeding pair anyways. And so at this scale, abundance is just not as biologically meaningful as occupancy. And so by using occupancy, it can also help us allow potential areas of breeding habitat. And so the habitat covariates that I'm looking at are these that I have listed. So stream flow, benthic substrate, forest type, stream character, which would be like, are there pools, are there loafing areas, backwaters, stream gradient, snowpack, and stream order. And I'm still trying to figure out if I can remotely sense these variables, especially at the scale I'm looking at. And so this is one of the current areas that I'm working on to figure out. But I do have corresponding hypotheses and predictions for each of these covariates. Okay. So this is a figure from the Strobel et al. Paper that I mentioned before. And so this figure is showing predicted occupancy for rock bunting in Switzerland using their integrated spatial distribution model. I plan on creating something similar for Harlequin ducks here in Western Montana and Northern Idaho. And so the steps I'm going to use to do that is I'm going to use my data type 1, so that repeated within season surveys, and the habitat covariates that we remotely sense to build that into my occupancy model. Then I'll use my data type 2, so the presence only, to inform the latent states of 1 if Harlequin ducks have been detected there. And then I'll use my absence only, so the augmented zeros, to inform my model of areas that we're really confident do not have Harlequin ducks in them. And so these last 2 steps of adding the presence only and the absence only data, they allow me to add reliable information about presence or absence between data type when data type 1 is not available. And so then I'll implement all of this in my integrated spatial distribution model using the JAGS language with covariates applied to occupancy state. And then predicted breeding season Harlequin duck occurrence on the remainder of the study area of stream reaches will be modeled as a function of the identified habitat covariate and their relationships. And I'll have one more field season in 2024. And so this field season will be used to address the data gaps that we may have. So this could include gaps in our covariate values. It could also address gaps in spatial gaps that we might have. And so to understand what these gaps are, I still am working through the data that I'm going to be receiving from both Montana and Idaho, and also figuring out what variables I can remotely sense at the scale that I'm interested in. And then I also aspire for the findings from my first chapter to inform what the field effort this upcoming season will look like for 2024. And with that, I have so many people I would like to thank. I would like to thank my advisors, Hannah and Josh Millspaw, I'd also like to thank Barb Lancaster at the Glacier National Park Conservancy for investing in me and for pushing for this project to be funded. My supervisor in Glacier, Lisa Bate, who's truly been one of the best mentors I could have asked for. Christina Smucker and Allison Bagley at Montana Fish, Wildlife and Parks, Casey McCormack at Idaho Fish and Game, Kara Staub with the Forest Service, Dan Bakken and Bryce Maxwell at the Montana Natural Heritage Program, all for providing me so much support over these last 2 years, and Peter Dudley from Montana Audubon for also providing support and helping me find people to help with these surveys, especially on the Rocky Mountain front. And so many more. I'd like to thank everyone in the Millspa lab, all of the technicians who have helped out over the last couple of years. My 2 technicians that I've worked with personally the last 2 years, Kai Martin and Megan Farrell. Yeah, there's so many more people I could put up here, all of you. So thank you. And I'll take any questions. And then, yeah.
It's really cool. One thing that I'm thinking about with the talk, I was wondering if you're planning on incorporating any of those habitat covariates, like for your second chapter, into the covariates that you're looking at in your first chapter, of like thinking about, I mean, just basic things like like terrain ruggedness or forest type, all that. Also, types of considering.
Yeah, that's a good question. I haven't thought a lot about that, but there's definitely a lot of overlap between the covariates that I'm including in both chapters. So a lot of the covariates that we collected in the field are also ones that we're like looking to see if we can remotely sense them. Yeah. Yeah.
With future questions, can you repeat it?
Oh yeah. Yes. Yeah, that's a great question. So the question for those of you online was for detection probability, if we're just sampling areas where we think there are Harlequin ducks, how do we think that can affect looking at like modeling areas that we don't know or don't have Harlequin ducks? And so that is a great question. I think for that, so the different streams that we chose them because they exhibit a variety of ecosystems and a variety of different stream characters. And so hopefully we were able to select streams that cover enough of the variety of these different stream systems. And then also because of that reason that the occupancy is 1, that's why our unit of replication is just 1 eDNA sample or 1 ground-based foot survey or 1 game camera, because not every single foot survey detected a Harlequin duck, or not every single eDNA sample detected them. And so that way we're able to narrow it down more to the detection process versus if they're there or not. Yeah. Questions? No.
I'm curious, thinking ahead a little bit. So from your occupancy, like,
Yeah, specifically Western Montana and Northern Idaho. Yeah.
you think from there,
Yeah, that's a great question. So the question was, looking at the occupancy model, do we think that we could use that to build a monitoring network or scheme? I think that like simple answer to that is yes. Yeah. Yeah, and so I think, you know, like the goal of this project is to provide like biologists that I'm working with and all of the people supporting this project with these tools of like, hey, here are the tools you have to detect them, and then here's this tool of like where they might be. If I had way more time, I would love to do that. And so see, yeah. Yeah. Any questions online, Cody? Okay. Sweet.
Thanks again.
Well, thank you, everyone. Yeah, this is a great training. Thanks, everyone, for coming.
Yeah, I get you.
Bye.
Bye, Holly. Good to see you.
Bye. Thanks. Good to see you. Thanks.
See ya. Way to go.
Thanks.
Good job, Molly. Good to see you.
Yeah, thank you. Good to see you too. Yeah, at 5. Oh yeah. Drinks at 5 if anyone wants to get killed. Yeah. Us, that's a 7. I planned it specifically because not everyone can fit in this. So yeah. Yeah. Yeah.
People are expecting. It'll say
Lisa, we have, I'm gonna close this down because we have to change rooms. And then I think there's that other link for the
I'm
closed
already
door.
on.
But
I'm
we'll,
already on the other link.
okay, we'll reconvene. I'll check with them. I know I have the room scheduled at 2:30, so you might
Oh,
have like
okay.
to take a break.
Okay. I'll hop back on. Good job.
Okay, sounds good.
Bye.
Okay, bye.